Good evening. So last week we did a video lecture on melanogenesis, which is one of the basic processes of dermatology, and I hope it was useful to people who actually watched it. And so I thought on the occasion of World Vitiligo Day, it becomes imperative that we discuss the pathophysiology of vitiligo. I've got multiple requests that we should discuss pathophysiology of diseases more. And what better day to discuss the pathophysiology of vitiligo when you have the World Vitiligo Day approaching on 25th. So, vitiligo is an important disease to understand. It is the most benign, devastating disease as per the patient, and it is very important for us to understand the nuances of how a vitiligo actually spreads or actually starts in a patient. For if we know better about the disease, it becomes easier for us to cure the disease, or at least counsel the patient, or at least help them to understand the disease process better. Okay, we can make it simply, we can simply explain it to the patient what exactly is happening, what is leading to the loss of pigmentation, loss of color in the skin, which is causing such an important cosmetic disfigurement to the patient. Now, always remember that vitiligo patients already suffer from the stigmatizing disorder of vitiligo, and they don't have the basic idea of immunity as well as we or any other doctor would know how immunity works. So it is our job, and in that it's especially on World Vitiligo Day, it is our job to make them understand what the disease process is. And if we want to explain it simpler terms to the patient, we must ourselves know why vitiligo actually happens. So that's the overarching aim of this video, that to understand basically how does vitiligo starts, or what are the initiating factors which will eventually lead to melanocytic destruction leading to vitiligo. So before you start this video lecture, I think one should always look at the last week's video on melanogenesis, because a lot of the factors, a lot of the players that we will learn about it today are already discussed last week in the process of melanogenesis. Okay, so all the genes, all the cell processes, all the cell intermediates, all have been covered in melanogenesis or the last week's video. And if you have gone through that video in detail, it will be a lot easier for you to understand the pathophysiology of vitiligo. Now, this video is quite extensive. It will cover nearly all points pertaining to the disease process leading to vitiligo. And if you go through this video once, I've tried to accumulate it from the three basic textbooks, Bolognia, Rooks, and Fitzpatrick, along with multiple good articles regarding the pathophysiology, and you will get a good in-depth idea about what all disease processes are there, and you can mention them while writing a short note on pathophysiology, or in multiple instances where residents might have their thesis on vitiligo, like I did during my junior residency. So it's better to understand deeply the causes of vitiligo, and when you know the causes, you can then think about how to treat the specific subset of vitiligo. So without much further ado, let's start our discussion on pathophysiology of vitiligo. So, we'll start with the definition of vitiligo. Okay, vitiligo is an acquired disorder characterized by circumscribed depigmented macules and patches that result from the loss of functional melanocytes. Now, this phrase, loss of functional melanocytes, is an important concept to understand, that the melanocytes are not able to carry their function properly. Okay, they're not able to form melanin, that leading to depigmented macules with well-defined borders, circumscribed, okay, well-circumscribed depigmented macules. So the definition of vitiligo is essentially it's an acquired genetic disorders characterized by circumscribed depigmented macules and patches resulting from the loss of functional melanocytes. And with this video, we'll understand what are factors are leading to loss of function in melanocytes. Now, the most important theory is that it is an autoimmune disorder, and that is explained by the fact that CD8 positive mediated cell destruction happens. Okay, CD8 positive T cells, they destroy the melanocytes, and because of destruction of melanocytes, you have depigmentation. The color is lost because the color forming cells are lost. And why they are lost? Because CD8 positive T cells have destroyed them. Okay. In the serums of vitiligo patients, anti-melanocyte antibodies have been formed. And these are antibodies against the antigens of melanogenesis products or the cell processes or any specific cell surface markers of melanocytes. And antibodies against them have been found in sera of vitiligo patients. Okay, so that makes this is as the most important hypothesis regarding pathophysiology of vitiligo. The second is abnormal melanocytes. That means the melanocytes by themselves are abnormal. So in autoimmune disorder, you have a normal melanocyte. Let's just make this like this, a dendritic melanocyte. And the melanocyte is okay, but T cells actually destroy it. So melanocyte is okay, but T cells destroy it. While in abnormal melanocyte, the melanocyte is not okay and dies by themselves. Okay. So these abnormal melanocytes have improper response to cellular stresses in vitro. That means the normal stress mechanisms that are happen because of any cellular process. See, cell undergoes a lot of processes. Okay, a lot of things, a lot of byproducts are created when the cell is alive. Okay, and that leads to formation of multiple reactive oxygen species. And these reactive oxygen species are handled by the antioxidant mechanism in a cell. But abnormal melanocytes are not able to handle that stress. And because of that, there is apoptosis of melanocytes. Okay. But always remember that vitiligo has a complex multifactorial hypothesis. That means not one theory is actually fully correct. At one point of time, in one individual, you can have multiple factors which are playing together to cause vitiligo. That is why the theory which is now the most popular is known as convergence theory. What do we mean by convergence theory? It means that there is abnormal, not abnormal, there is, let me write it again, there is non-functional melanocytes. Okay, and this is the crux of vitiligo. So they could be autoimmune destruction, or they could be abnormal melanocytes, or they could be reactive oxygen species damage, or they could be UPR mediated damage. We'll discuss that in detail, don't worry. Any kind of stress leading to damage. So all of these damaging processes converge on non-functional melanocytes. Okay. And that is why it is known as convergence theory. That means multiple factors are coming together to cause non-functional melanocytes. Now, abnormal melanocytes usually secrete reactive oxygen species in a lot more quantity, okay, along with multiple unfolded proteins. Now, you have to understand that whenever a cell undergoes any kind of stress, the normal protein folding mechanism is altered and the cell is not able to fold protein properly. And because of that, there is accumulation of unfolded and misfolded proteins. And when these proteins accumulate, it creates a sort of danger signals and it signals the innate immunity to come and help. For example, if a cell is not working properly and a lot of misfolded proteins are accumulated, it will signal the innate immunity to either destroy the cell or correct the damage. Okay. Now, this innate immunity, it activates the CD8 positive T cells, and these activated T cells destroy abnormal melanocytes. So this is the major process which is happening to cause vitiligo. The third is chemical theory, in which phenols compete with tyrosine and produce cellular stresses. Now, as you know from last week's video, that tyrosine was a starting molecule in melanogenesis, and it is a very pro-oxidant molecule. It's a very pro-oxidant molecule because if you remember the process, we were dealing with quinone molecules, hydroxyl molecules. So a lot of nascent oxygen is being created while the cell is forming melanin. Okay, and because of that, as a melanocyte requires good processes to limit the formation of nascent oxygen. Okay, if the melanocyte is abnormal, you will have accumulation of nascent oxygen, and because of that, the cell is susceptible to ROS-mediated damage. Now, there are other chemicals which are also acting like tyrosine to form a lot of oxidative species, and phenol is one example. That is why you have phenol-mediated depigmentation. People who are allergic to phenol gloves can have depigmentation of hands because of that. Okay. The fourth is neural hypothesis. So in the last week's video, we had discussed in the video on melanogenesis that the melanocytes are derived from neural crest cells, from the crest or the neural tube. And they go through the dorsolateral pathway majorly and minorly by the ventromedial pathway. And during the process of migration, by the action of multiple melanocyte growth factors, inhibitors, and all sorts of molecules, the melanocyte stem cells or melanoblast stem cells gradually gets converted into adult melanocytes. And this migration is specific to dermatomes or neurotomes. Okay. And if there's a problem in the migratory pathways, you will have defective melanocytes reaching the end stage in the skin. And if a pathway is defective, the entire area supplied by those melanocytes can be defective, leading to segmental appearance of vitiligo lesions. Okay, so neural hypothesis is mainly in the context of segmental vitiligo. Segmental vitiligo, where a certain area part of the body, well circumscribed, is involved with vitiligo patches. Okay, but there's a lot of conflicting evidence with this, and that is why it's considered as a weak hypothesis. Okay, let's move forward. So this diagram highlights the intrinsic abnormalities in vitiligo. Okay, I'll just give you some time while I'm talking. You can take a screenshot of this area. So we know that melanocytes, or at least the abnormal melanocytes in vitiligo, they increase the production of reactive oxygen species. The antioxidant mechanisms to counteract this ROS production is defective. There are mitochondrial alterations, and mitochondrial alterations lead to defective antioxidant mechanism. They also lead to generation of reactive oxygen species, and they also lead to apoptosis, melanocyte apoptosis. Okay. The keratinocytes, the surrounding keratinocytes, they secrete TNF-alpha and interleukin-1. Remember in the pathophysiology of psoriasis video, we discussed that keratinocytes are a good source of TNF-alpha. And increased TNF-alpha will lead to inflammation. Okay? And increased inflammation means increased inflammatory damage to the melanocyte, increased recruitment of T cells towards melanocytes. There will be more destruction of melanocytes. So decreased stromal cell factor, which is actually a good growth factor, that is decreased. Bacterial degeneration, swollen mitochondria. Swollen mitochondria is a sign of oxidative stress in a cell, and if it is not corrected quickly, the cell might go under apoptosis. NRLP3 activation or the inflammasome pathway is a newer activation. Essentially, it means that there is activation of an organelle known as inflammasome, which leads to different pathways of cell death like apoptosis, necroptosis, ferroptosis. And this is just to have a clear understanding. Just remember that keratinocyte increases inflammation. Defective melanocyte increases oxygen, reactive oxygen species, and fibroblast increases stress on the melanocytes. Just remember these three things and you'll be easily able to understand. And the stress is mainly by reactive oxygen species. So fibroblasts increase ROS, melanocyte increase ROS. They are not able to battle with ROS because the antioxidant mechanisms are not good. And keratinocytes increase inflammation and inflammatory chemotaxis of cells. Clear? Just understand this. Let's move forward. Now this diagram essentially shows the same thing. You have the fibroblasts. Okay, these are all parameters that we will discuss in detail while going forward with this video. Now damaged melanocyte will release some molecules which are known as DAMPs. DAMPs are damage associated microbial proteins. These are just you could say pieces of cellular material which when released into the circulation alerts the innate immunity that something is wrong. Okay, it's like a calling card, it's like an alarm bell. So when a melanocyte dies, mainly because of a stressor, which is the trigger event. Okay, trigger event, which could be anything, um, chemical trauma. Okay, so this trigger event, when this trigger event acts on a melanocyte, which is abnormal, which is genetically susceptible, it will die and release the cellular material. When cellular material is released, it is recognized by dendritic cells. And after recognition, these cells gets activated and they present these cells to circulating T cells. And when these T cells are circulated, they get activated because DAMPs are a marker of cell death. So just get this concept very clear. Abnormal melanocyte under the action of stressors will release DAMPs. DAMPs go to dendritic cells, activate them. The dendritic cells act as antigen presenting cells and activate circulating T cells. And these circulating T cells will go to the skin and destroy melanocytes, and further melanocytic destruction happens. Okay, so that is how the problem with vitiligo starts. Now look at this arm. Look at this arm. You have TRM. TRM is nothing but resident memory T cells, and they are responsible for relapses in vitiligo. We'll discuss resident memory T cells in this video subsequently. Let's move forward. Okay. So we have gone through an overview of the pathophysiology in vitiligo essentially. Now we will go deeper into each of the individual hypotheses and understand what exactly is happening. Okay. Uh, just a second. Yeah. Yeah, what exactly is happening? We'll understand that. We'll start first with the autoimmune hypothesis, which is the most important hypothesis. Most important doesn't mean that only it's the only major hypothesis. It is just that in the convergence theory, it is the main arm, okay, main pathway which is causing destruction of melanocytes. Now, there are autoreactive CD8 positive T cells which are found in lesional and perilesional skin in a vitiligo patient. That means the destruction is carried on by majorly CD8 positive T cells which act against melanocytic antigens. Okay. Antigens include tyrosinase, which is the major antigen as of now, as far as studies are concerned, major antigen in vitiligo. Other antigens include melan-A, MART1, GP100, TRP1 and TRP2, which are essential molecules catalyzing the process of melanogenesis. Okay, last week video we discussed them in detail. Now, the autoreactive CD8 positive T cells are activated because of major autoantigens. Most important of them is tyrosinase, and this leads to melanocyte destruction. And when melanocytes are destroyed, we see that in the form of lack of DOPA positive melanocytes in stratum base cell. So DOPA positive are actually it's actually a marker for melanocytes. Remember, in melanogenesis, tyrosine leads to L-DOPA formation. So if the cells have DOPA, they are producing melanin. They will produce melanin. Okay. And you don't see dopa positive in stratum basal cell. That means those cells are actually destroyed. Okay. So here we have a dendritic cell, this cute little stupid blue cell. That's a dendritic cell. And on the activation by melanocytic antigens, okay, any autoantigens, it leads to activation and increased stimulation of a naive T cell. So this is a naive T cell. It is just in circulation and it is just biding its time. But upon activation by a dendritic cell, which acts as an antigen presenting cell, this naive T cell gets activated to form an angry autoreactive T cell. Autoreactive means it will act against the body, reactive against the body, and this autoreactive T cell, CD8 positive T cell, would eventually destroy melanocytes. So there's an inherited genetic risk. That means there is a genetic component with vitiligo also. Remember, vitiligo is a multifactorial disease. Okay. So genetic risk and environmental insults and stresses could be trauma, could be chemical, could be anything. This increases melanocyte stress. So a melanocyte which is genetically susceptible to not able to handle stress, and the stress is applied by the environment to the cell, it increases this melanocyte stress which the cell is not able to handle, and this increased stress of the melanocyte leads to a cascade of events which eventually activates the T cell. Okay, so when melanocyte stress is increased, it signals to local innate and resident skin cells via exosomes. So exosomes is nothing. You have a cell and you have a little vesicle which has all the material, microRNA, polypeptides and all. And this, when attached to the cell membrane, is secreted out. And this is what is exosomes. These are important for cellular communication between cells. Okay. And using exosomes, of course, there are multiple pathways available. One of the pathways exosomes. Using exosomes, the damage or the under stress melanocyte signals to local immune cells by releasing antigens and damage associated microbial proteins, which and heat shock protein 70, which is a very important DAMP. And when these are secreted, they activate the T cells. They stimulate the autoreactive T cells. And when T cell is autoreactive, it increases interferon gamma. And this is the main mediator of vitiligo. So remember the main mediator of psoriasis was TNF alpha. In vitiligo, it is interferon gamma. Let's move forward. So interferon gamma, okay? Interferon gamma attaches to interferon gamma receptor on keratinocytes and other cells surrounding the melanocyte. And by the action of STAT1 pathway or the JAK-STAT pathway, JAK1 and 2, it phosphorylates the STAT1. And when STAT1 is phosphorylated, it goes to the nucleus and attaches to the gene. Okay, so what actually happens is you have interferon, it attaches to the receptor, and this leads to activation of the JAK-STAT pathway. When JAK-STAT1 and 2 is activated, it goes inside and reaches the genetic material in the nucleus. And when it attaches itself to genetic material, it leads to formation of multiple genes, mainly CXCL10, CXCL9. And when these two are increased, they will act on T cells which have CXCR3 receptor. These two, when they act on this receptor, they will stimulate this T cell and convert it into an autoreactive T cell. So that is how the T cell, when the T cell gets activated, it leads to formation of interferon gamma, which leads to the formation of STAT1 pathway, which leads to the increased transcription of CXCL10 and CXCL9, which when they act on CXCR3 receptor, it activates the autoreactive T cell, and this activated T cell will go to the skin and where it will damage the melanocytes. Damage melanocytes. Now let me recap what exactly is happening. Okay, listen to me carefully. There's an abnormal melanocyte which, under the action of environmental stress, releases exosomes, DAMPs, HSP70. On the release of these molecules, the dendritic cells gets activated, and when the dendritic cells are activated, it's going to go and activate the naive T cell. When the naive T cell is activated, the naive T cell converts to an autoreactive T cell. Now, this autoreactive T cell will secrete interferon gamma, and this interferon gamma will act on the interferon gamma receptor on keratinocytes. On reaching the keratinocytes, the interferon gamma will activate JAK-STAT1 and 2 pathway. When that pathway is activated, it will homodimerize and go to the nucleus and increase the transcription of CXCL10, CXCL9. And when CXCL10 and 9 are secreted, they will act on CXCR3 positive T cells, which are the autoreactive T cells, and home them or call them towards the skin, where they will eventually go and damage the melanocytes. So now the whole process is clear. I hope it is clear. If it's not, mention in the comment section and I'll personally answer any queries. So all thanks to autoreactive T cell. Let's move forward. So quickly, with this diagram, which I've taken from Bolognia, page 1088, we will recapitulate the autoimmune theory of vitiligo pathogenesis. So melanocyte stress, mainly because through the environment, along with intrinsic risk factors in an abnormal susceptible melanocyte. Okay, this whole brown blob that you are seeing is actually a melanocyte. Okay, and because of intrinsic risk and melanocyte stress, mainly because of the environment and the genetic risk. It leads to increased production of reactive oxygen species. Now, be with me. I'll tell you how it is not an oxidative mechanism, but more of an autoimmune mechanism as of now. The tyrosinase enzyme and the TYRP1, which is responsible for melanogenesis, also makes more reactive oxygen species. Now, increase in reactive oxygen species will lead to endoplasmic reticulum stress because of unfolded proteins, misfolded proteins, and all. Okay, everything causes destruction of melanocytes. When the melanocytes are destroyed, they will secrete HSP, heat shock protein 70, and via exosomes, HSP 70 and other damage associated microbial peptides are released into the circulation. When these are released, they will go to the dendritic cells and activate them. When the dendritic cells are activated, they will activate the migrating cells and the resident cells. Okay, the resident cells are there, which are in the nearby environment, and the activated cells are those which are in the circulation. Now, these activated cells, sorry, the cells in the circulation under the activation and stimulation by dendritic cell will get converted into activated T cells. Now, activated T cells will form interferon gamma. Interferon gamma will act on the receptor on the keratinocyte. This is a keratinocyte, and keratinocyte, by the action of JAK-STAT1 and 2 pathway, will secrete CXCL10. This CXCL10 along with CXCL9 will act on CXCR3 autoreactive CD8 positive cell. And when CXCL10 attaches to CXCR3, it will home the cell, or in other words, homing will happen. The cell will be called to the skin, and at the level of skin, the cell will come and destroy the melanocytes. So remember, melanocyte under stress releases chemicals. Those chemicals activate dendritic cells. Dendritic cells activate T cells. T cell release interferon gamma. Interferon gamma activates CXCR3 positive T cells, which go to the skin and destroy the melanocytes. Clear? Let's move forward. So in the previous few slides, we have discussed what is the normal mechanism or the normal pathway as of now, as per the different article studies, leads to autoimmune mediated damage of melanocytes. Now we'll talk a little bit about resident memory T cells. These are those specific type of CD8 positive T cells that express markers like CD69, CD103, CD49A. Okay, they're known as resident because they remain in tissues. So as resident memory T cells remain in skin. Okay, they stay there. And while staying there, they can induce early immune response. Why? Because they are closest to the damage. Closest to skin, closest to damage. These resident T cells will release interferon gamma. And this interferon gamma will activate T cells. We have already discussed how interferon gamma actually activates T cells. So if you have a resident T cell which stays there, it will continue to release interferon gamma and under the action of any stress, any stimulation, and this will further activate T cells, and that is why you have repeated relapses in vitiligo. You understand the concept? Let's say there is some kind of riot going on in a city, okay, and the police commissioner comes and all the firing happens, they made sure that all the rioters are caught. After everything is done, the police commissioner will put one sipahi at the junction and says to them, if anything happens, call me back. That sipahi is resident memory T cell. That police commissioner is CD8 positive reactive autoreactive T cell. Let's say after the riot has been subdued, some miscreants come and start causing riots or try to cause riots again. The sepoy will immediately call the activated T cell or the police commissioner. The police commissioner will come again and the whole process of subduing the riot happens again. Similarly, the resident memory T cell will keep on calling the T cells upon activation, and this repeated calling of T cells are the one responsible for relapses that we frequently see in vitiligo patient. I hope this metaphor makes it clear. Okay. Interleukin 15 and interleukin 7 are responsible for maintenance and function of this resident memory T cells. So when you are having increased secretion of 15 and 7 interleukins, the resident T cells are present and they are activated or they are constantly on the lookout for fresh damage leading to relapses. So if you somehow block these cytokines, you might block relapses in vitiligo. Let's move forward. So, in the previous slides, we have discussed about the autoimmune mediated damage to melanocytes. Now, let's discuss the second theory, the oxidative stress. Okay, this is the second most important theory in vitiligo pathogenesis. So, let's start. Oxidative stress is regarded as a trigger event. That means the event that actually starts melanocytic destruction. And in the previous slides, we have discussed that abnormal melanocytes under stress will produce a lot of reactive oxygen species leading to increased oxidative damage to melanocytes. Okay, vitiligo melanocytes are more susceptible for oxidative stresses. Melanocytes, by the sheer production of melanin, produce reactive oxygen species. This is normally seen in a normal melanocytes. Okay, normal melanocyte will form reactive oxygen species, but a normal, good functioning melanocyte has mechanisms to counteract that oxidative stress. But if the melanocyte is abnormal, that antioxidant mechanism doesn't work properly, leading to increased buildup of oxidative stress, which eventually damages the melanocytes. We know that melanogenesis is a pro-oxidative state. That means there's production of more ROS when the melanin is also produced. This is normal. Now, increased ROS, when reactive oxygen species are increased, it leads to structural damage to DNA, proteins, lipids, cell organelles, and mitochondria. And if these kind of cell organelles are damaged, the cell is pushed more towards apoptosis. And we all know what is going to happen. After apoptosis, you will have secretion of DAMPs, which will activate dendritic cells, T cells, CXCR3 T cells, eventually leading to more melanocytic destruction. And that is how oxidative stress is considered as a trigger, and the autoimmune pathology is the main branch through which the melanocytic destruction actually happens. Let's move forward. Let's talk a bit about protective machinery. What actually prevents the buildup of oxidative stress in a normal melanocyte? Now, whenever the cell is under stress, the normal protein folding doesn't happen. And when the protein folding is altered, there is accumulation of defective peptides, accumulation of defective proteins. Now, proteins are found by smaller units which are known as peptides. When they're not able to attach themselves together in a proper manner because the cell is under stress, it leads to multiple wrongly folded protein. And when that happens, there is accumulation of misfolded proteins. When this misfolded proteins goes inside endoplasmic reticulum, now endoplasmic reticulum is responsible for packaging and transport of proteins where the proteins are abnormal. So endoplasmic reticulum will swell. There will be swelling of ER. And this swelling of ER starts a cellular stress phenomena known as unfolded protein response or UPR. And this is a marker of cellular stress. Okay. There's also increased CGRP, which is calcitonin gene-related peptide, and TRPM2, which is transient receptor potential cation channel subfamily M member 2. You don't have to remember anything, just remember that increased cell stress leads to increased UPR, increased CGRP, increased TRPM2, leading to melanocyte apoptosis. And we all know what happens after apoptosis, autoimmune pathway, destruction of melanocytes. Okay. Now, there are protective pathways against these active oxygen species, but in abnormal melanocyte, these are defective. Let's move forward. So in the previous slide, we had discussed that there is increased ROS leading to increased unfolded protein response in stress keratinocytes and melanocytes. There's increased CXCL16 and this CXCL, remember CXCL are chemokines, okay? They're responsible for chemotaxis. So CXCL16 will increase the recruitment of CD8 positive T cells. And what happens when CD8 positive T cell reaches melanocyte? Melanocytic destruction, which is vitiligo, clinically known as vitiligo. Clear? The concept is clear? Now, reactive oxygen species also decrease WNT expression. And remember, WNT expression is responsible for good migration of melanocytes. So when melanocytes are getting migrated from neural crest cells to their eventual location in the skin, WNT expression is important. Okay. And when WNT expression is altered or decreased, this migration doesn't happen, this differentiation doesn't happen. And because of that, they are abnormal melanocytes. Clear? And this WNT expression is decreased by reactive oxygen species. Chronic mechanical stimulation of skin also increases the active oxygen species, and that is the implication or that is the explanation given for commonest phenomena. Remember, in active vitiligo, wherever there is skin trauma, the depigmentation happens. That is known as commonest phenomena. Why is it because of that? That chronic mechanical stimulation or chronic mechanical trauma increases reactive oxygen species, and this ROS kickstarts the pathway of melanocytic destruction, leading to commonest phenomena. Okay, let's move forward. So in oxidative stress, there's a concept of oxidant-antioxidant balance. That means a balance has to be maintained. Remember, a normal melanocyte will always form oxidant-free radicals by the process of melanogenesis. But the cell also has mechanisms to counteract it. When the mechanisms fail to counteract it, you have oxidative damage. Glutathione is a first line of defense. It's a very important antioxidant. So what actually happens is GSH is responsible for the donation of hydrogen atoms. So you have two molecules of GSH which will donate H2. Okay, they will donate H2 and form GS double bond G. Okay, and this H2 will add an oxygen free radical and will form eventually H2O. Okay, that is how glutathione scavenges oxygen free radicals. But there is a mechanism to convert GSSG back to GSH, and that is done by glutathione reductase. And this glutathione reductase under stressful condition is inactivated. And when this is inactivated, GSH is not formed properly. And since GSH is not formed properly, it is the first line of defense, and that line is now broken. And that is how the balance shifts towards antioxidant preservation to oxidative damage. Clear? So in vitiligo, enzymes like glutathione reductase, glutathione peroxidase, catalase, methionine sulfoxide reductase, these all enzymes which are responsible for keeping the oxidant-antioxidant balance, they are all defective. They are not able to work properly, and as a result, the oxidative stress increases. So remember, glutathione is a first line of defense which is not working properly in patients of vitiligo. It has been found in multiple articles, and the melanocyte is eventually not able to handle the oxidative stress. Let's move forward. So these cells, there is defective mitochondrial function. And remember, mitochondria is the area where the signal for apoptosis begins. Whenever the cell is undergoing any kind of damage, there's signal of mitochondria to multiple proteins like BCL2, BX, BAX, BAM-bit proteins. All these proteins activate the mitochondria. When the mitochondria is not functioning properly, the cell is not able to form its ATP, and because of that, the cell is now programmed for apoptosis. The cell is useless, we have to get rid of this, and that is how apoptosis starts. Okay. So all the enzymes that we have discussed previously, superoxide dismutase, glutathione peroxidase, glutathione reductase, all are defective in maintaining the proper oxidant-antioxidant balance, and the balance shifts towards more oxidative stress, leading to increased ROS production and damage to melanocytes. When the melanocytes are damaged, the antigens inside the cells are unveiled, they are released into the circulation, and these antigens, these autoantigens, will activate T cells, and these T cells will lead to further death of melanocytes, leading to vitiligo. Clear? Let's move forward. Now let us discuss in detail what exactly is this unfolded protein response in the endoplasmic reticulum. So on the previous slides, we had discussed that UPR is increased when ROS is increased. When reactive oxygen species are increased, unfolded protein increases. Okay. So whenever there's increased oxidative stress, the cell proteins are damaged. When these proteins are damaged, the endoplasmic reticulum is not able to function properly because the cell has undergone damage. The main function of ER and Golgi apparatus is to process the protein secretion of the cell. but the cell is damaged. So what happens is unfolded proteins start to accumulate. They start to increase in cell, and that triggers unfolded protein response. It is a newer concept in vitiligo pathogenesis. So normally, when there is unfolded protein response, a signal is sent to the cell that you should restore ER function. Increase UPR means that endoplasmic reticulum is not functioning properly. We need to correct it. That is normal function of UPR. But when UPR is increased to that extent that it crosses a threshold, the endoplasmic reticulum starts to swell. And the swelling of organelle is a sign of cell undergoing apoptosis. So whenever organelles like endoplasmic reticulum or mitochondria start swelling, remember the cell is going to go under apoptosis. And that is how the cell undergoes apoptosis because of unfolded protein response leading to melanocyte destruction. And we all know what happens, T cell activation. Let's move forward. So there's in the response to unfolded protein, you have increased interleukin 6, interleukin 8, CXC ligand, and these all ligands and interleukins are responsible for increased CD8 positive T cell migration. And this is the villain of the story. Let's just say it's the villain of the story, which will destroy melanocytes. The unfolded protein response also increases the pro-inflammatory response in macrophages. So macrophages, another part of innate immunity, which when activates will destroy cells, and that is activated or a pro-inflammatory response is started in because of unfolded protein responses. Okay, let's move forward. Exosome signaling. So we have previously also talked a little bit about exosomes. They are just responsible for cell-to-cell communication. So exosomes are mediators of communication between stress melanocytes and the innate immune system. So it's like a radio channel between a damaged melanocyte and an innate immune system. Any stress leads to secretion of exosomes that contain microRNAs, melanocyte-specific antigens, heat shock proteins, and other proteins, which are also known as damage-associated microbial proteins. So what actually is happening by using exosomes, a damaged melanocyte is sending signals to innate immunity that there is something wrong with me. You should come and have a look. So that is how what essentially is happening. These exosomes, when they reach dendritic cells, they stimulate maturation into antigen-presenting cells. And this leads to T cell stimulation, which is nothing but a shortened form of autoimmune pathway. Autoimmune pathway. So what exactly is happening? Exosomes are released whenever the melanocyte is under stress, and it goes to the innate immunity and activates dendritic cells. Activated dendritic cells form good antigen-presenting cells. These APCs then further stimulate T cells. Let's move forward. So this diagram summarizes all these concepts that we have talked about. We know endoplasmic reticulum stress increases unfolded protein response. These are the 6 and 8 interleukins, and CXCL 9, 10, and 16 are released. These are responsible for recruitment of CD8 positive T cell. Recruitment of CD8 positive T cells. Heat shock protein. Now, this is a very good marker or a very novel marker in the pathomechanism of vitiligo. Okay, so heat shock proteins, which is actually a form of damage-associated microbial peptides or proteins, they activate dendritic cell, and dendritic cell will activate further activated T cells. Heat shock protein can eventually also directly activate T cells. That somewhere damage is going on, and you should have a look. Exosomes also, because as I've already mentioned, that they might contain microRNAs and autoantigens, and this autoantigen also activate T cells. And if the cell dies or undergoes apoptosis, they can also release the autoantigens into circulation, further activating T cells. Let's move forward. Okay. Now here, this is just to recapitulate that chemical triggers can also act as a source of increased cell stress. And when the cell is stressed, the reactive oxygen species increase, UPR is activated. Autophagy means that the cell starts to digest its own organelles. Now, what exactly happens is, let's say I'm a cell, and I have to send a signal to innate immunity that something is wrong with me. You need to come and destroy me. What I will do is I will digest my own organelles and will present that pieces of organelles to the dendritic cells, which will get activated because they sense that something is wrong. So these species are known as damage-associated microbial peptides, and by the activation of various factors, recruitment of macrophages, activation of inflammasomes will lead to cell death. So all the processes that we have discussed so far is eventually leading to keratinocyte death. Just remember that. Since everything is converging into melanocyte death, this is known as convergence theory. Okay, let's move forward. Okay, so we have discussed two major pathways of melanocyte damage. We have discussed autoimmune mediated damage. We have discussed oxidative stress triggering event for melanocytic damage. The third important theory is melanocytorrhagy, in which the melanocyte loses its attachment to the surrounding keratinocytes. Okay, remember that in the autoinflammatory hypothesis, there's increase majorly in interferon gamma, majorly, and also via keratinocytes, there is increase in TNF-alpha, keratinocytes. Remember that in the first, I think, third or fourth slide we were discussing. Now, all of this increased TNF-alpha and increased interferon gamma releases matrix metalloproteinases 9. And metalloproteinases, what they do is they destroy the cellular stromal material. So what actually is that you have all the cells, and in between the cells you have stroma, stroma, okay, stroma, and MMPs, matrix metalloproteinases, destroy stroma. So MMP9 is increased in response to TNF alpha and interferon gamma. Also, Dickoff related protein 1, which is secreted by fibroblast. They secrete DKK1. It's an inhibitor of WNT signaling. Okay. So DKK1 is overexpressed in the dermis of vitiligo. Where exactly are fibroblasts? They are at the dermal level, dermis. They secrete DKK1. DKK1 is an inhibitor of WNT, and we know WNT is responsible for proper migration. If WNT is inhibited, migration doesn't happen properly, and the cells are not able to form properly, and they reach their place. ICAM1, or intracellular adhesion molecule 1, is responsible for attachment of T cell and melanocyte together. And ICAM1 is increased in vitiligo. So what ICAM does is, here you have a T cell and here you have a dendritic melanocyte. ICAM1 is responsible for attachment of these two cells together, and when ICAM1 is increased in vitiligo, there will be more attachment of these two cells and the T cell will be able to properly destroy keratinocytes. So that is how increased ICAM1 leads to destruction of melanocytes. The fourth theory, or the important theory, nowadays it is a weak hypothesis, it's not that strong, is known as neural theory. Now, neural theory was initially considered a very important theory, mainly in segmental vitiligo or symmetrical non-segmental vitiligo, or vitiligo seen after nerve injury. So it was said that because the nerves are the migration of nerves is in a dermatomal distribution in adult body, and in segmental vitiligo you have isolated, well-defined segments of the body involved. That means the migration of nerves are somehow affecting melanocytic activity. So that's how this theory came together. So neuropeptides like neuropeptide Y is seen increase in lesional and pre-lesional skin. These are all the data that I've taken from various articles that what all peptides are increased. Substance P is increase in skin of stable vitiligo. That means it is directly proportional to stability. Catecholamines are increased in the lesional skin, and catecholamines actually increase reactive oxygen species and immune dysregulation. Okay, so all of these factors was found to be increased in the local milieu in segmental vitiligo. Also dopamine. Remember, dopa is one of the mediators, or one of the middle molecules in melanogenesis. And dopamine is the most potent toxic monoamine to melanocytes. So it's a toxic monoamine, it damages the cell. Okay. So in the neural theory, it was said that the melanocytes that are directly or indirectly supplied by nerve trunks, whenever there's injury to nerve trunk, the melanocytes are not able to survive and they undergo depigmentation. Okay. And the migration of melanocyte is similar to the growth of nerve cells, and that is why you have segmental involvement in segmental vitiligo. So initially it was thought that it was more of a dermatomal or a multidermatoma involvement, but gradually it was found that multiple different segments also exist in vitiligo which are not dermatomal. That is why this theory is now considered a bit weak, and that's why we're discussing it at number 4. But you have to mention because it is an important theory, especially as far as segmental vitiligo, symmetrical non-segmental vitiligo, or vitiligo after nerve injury is concerned. Clear? If you have any doubt in understanding neural theory, write down in the comments and I'll answer them. Let's move forward. Now, the fifth theory is somewhat of a latest theory, and I'll keep this fifth theory segment, especially for the members, somatic cutaneous mosaicism theory. So this is a further improvement in the neural theory pathway. And this theory came about because of segmental and symmetrical involvement that we see in segmental vitiligo and non-segmental vitiligo. That in segmental vitiligo, the area involved might not be dermatomal, so it cannot be attributed mainly to the neural pathways. And there are multiple restricted areas of depigmentation. For example, pigmentation of the nipple areola complex. In that area, you don't have a dermatome which supplies only to the nipple areola, or a dermatome that supplies only around the mouth, or a dermatome that supplies only on the ear, or the central part of face which is involved, or the genitalia which is involved. But these are the areas which are involved in segmental vitiligo and non-segmental vitiligo more commonly than the other areas. So it was theorized that there are certain distribution patterns of melanocytes, and if any problem occurs in one of the pathways or any of the distribution pathways, the melanocytes are not able to attach properly to surrounding cells, leading to defective melanocytes. And this leads to melanocytorrhagy, or we have already discussed before, the damage to melanocytes. Okay. So in short, it is a further improvement into the neural theory of vitiligo that there are certain restricted areas which are supplied by one clonal cells of melanoblast, which if they are defective can lead to segmental vitiligo. And this is a bit separate from Blaschko lines. How is it separate from Blaschko lines? Blaschko lines are mainly involved with epidermal disorders or epidermal growth disorders like epidermal nevi, epidermal growth nevi, melanocytic nevi, mainly nevus disorders. Okay, while vitiligo is more of a pigmentary disorders. In Blaschko, the keratinocytes are affected more. In vitiligo or segmental vitiligo, the melanocytes are affected more, and that is why this somatic cutaneous mosaicism theory is a bit different from the mosaic disorders of Blaschko. Okay. But it is also observed that vitiligo can appear at all stages and all ages. It can appear at 2 years, it can appear at 20 years, it can appear at 80 years. And that is because damage can happen at various stages of migration and development. And whenever the cell is damaged, you will have induction of vitiligo in genetically susceptible individuals. So I will recommend to read this article by Atelier VR. I'll put that in the description. So you can go and ahead and read it. It's an interesting article. So let's in next 3 slides we will just combine everything that we have learned about the pathophysiology of vitiligo. Okay, so about 80% are genetic factors, about 20% are environmental factors. Each of these factors lead to stress inside the melanocytes. Okay, now these melanocytes under stress will produce a lot of reactive oxygen species. This reactive oxygen species will alter membrane lipids, leading to separation of melanocytes from keratinocytes. These reactive oxygen species will also decrease E-cadherin. E-cadherin is responsible for attachment. If that is decreased, the cell will lose its attachment. This stress will release unfolded protein responses, which will lead to CXCL16 majorly. The reactive oxidant species will lead to formation of exosomes as a signaling molecule to dendritic cells that something is wrong. When the dendritic cell is activated, it will present those antigen to T cells. The unfolded protein response is also activating T cells via CXCL16. Eventually, the T cell gets activated. And since the homing molecules like CXCL16, CXCL10, CXCL9 is happening, the cell will now go to the skin. This is known as initiation in vitiligo. Let's move forward. Yeah. So we had initiated vitiligo in the last slide. Let's let's progress it. Progression is mainly that the CD8 cell has now reached skin. Okay. On reaching skin, it will act through HLA polymorphic receptors or HLA receptors and will cause the apoptosis or the destruction of melanocytes. When the melanocytes are destroyed, it will release more damage-associated membrane proteins. It will release more of HSP70. It will release more of exosomes and all the other signaling molecules, leading to more activation and recruitment of activated CD cells. And when more and more immunity cells are coming here, there will be increased progression of it. Like clear. So in first step, the cell undergoes stress-mediated destruction, leading to activation of T cells. In the second phase or the progressive phase, increased activation of T cells will lead to more cell destruction, and that is how vitiligo spreads. The third part is maintenance, and maintenance is by resident memory T cell, which under the action of interleukin 15, CD122 will keep on activating the T cells to form reactive T cells, which will come and destroy keratinocytes. Okay. Now, resident memory T cells will also they release interferon gamma. We know that we have discussed in previous slides, and this interferon gamma, by the action of JAK pathway, will cause the keratinocytes to secrete molecules like TNF alpha. And what is TNF alpha? TNF alpha means inflammation. And inflammation means inflammatory mediators will come and attack and kill keratinocytes and melanocytes. Okay, that is why in many of the articles it is mentioned that biopsy of vitiligo shows inflammatory mediated damage. Clear? So I hope that in the last three slides we have quickly summarized what is the pathomechanism of vitiligo. So with that, I finished this humongous video. I had to shoot it without a fan and AC, so that's why I'm sweating a lot. So without much further ado, I would like to stop. It's very dehydrating now, shooting in this weather. Read Fitzpatrick, ninth edition, chapter 76 for vitiligo, chapter 66 in Bologna's 4th edition. This article, an update on pathogenesis, it's a good article if you want to understand genetic basis. If you want to understand the oxidative stress, then this article by Chang WL is very good. Go through it. Zai article on the endoplasmic reticulum dysfunction or the unfolded protein response is good if you want to read that in detail. Vitiligo mechanism of pathogenesis treatment focuses more on autoimmune damage. So if you want to go deeper into that, you may read this. I have not used Rooks much. Rooks is not that good as in this particular scenario. It is not good in explaining vitiligo, so I have not used that much. But these individual articles are good. If you search for better and newer, recent articles, latest articles, there's a plethora of new research going on in vitiligo. Let's move forward. So with that, we finished the discussion on pathophysiology of vitiligo, and on the occasion of upcoming vitiligo day. I thought it's a good time to discuss about vitiligo. All the genetic control, the autoimmune mediated damage, the oxidative stress mechanisms, and the melanocytic apoptosis, everything has been discussed in detail. If you go through this 1 hour video lecture, I hope that your understanding about pathomechanism of vitiligo actually improves and you're able to make sense of what actually is happening. And you should not say to patients that it is just an autoimmune disorder. No, it's not just an autoimmune disorder. Multiple factors are there. You'll be able to explain them then that because of a certain destruction of the cells, the immune system is now regarding those cells as foreign. They're going to continuously getting destroyed by activated T cells, and we'll do our best to stop this antigen or autoreactive T cell mediated damage. And that is why they need different immunosuppressive drugs or JAK-STAT inhibitors or narrowband UVB, whatever it is, to actually they can easily understand what actually is happening. Okay, so I won't take much of your time, even I am tired and dehydrated. Thank you for listening. You may comment any doubts, suggestions, or queries in the comment section. I do answer them. Okay, or you can directly email me at this ID. I will not take much of a further time. I need to drink some water. Adios. Bye bye. And enjoy your weekend. Bye.